Good morning. It's September 8th, and this is your daily brief in science. Here's everything you need to know. Russian health officials have announced that the Enteromics mRNA-based cancer vaccine has successfully completed preclinical trials and is now ready for Phase 1 clinical trials. This development marks a notable step forward in cancer treatment research. The vaccine was developed by the National Medical Research Radiological Center and the Engelhardt Institute of Molecular Biology. It is designed to target and destroy cancer cells, shrink tumors, and is personalized for each patient. In initial clinical trials involving 48 volunteers, the vaccine showed promising results, including slowing tumor growth and even complete elimination of some cancers. The primary focus initially is on colorectal cancer, but ongoing research aims to expand its use to other aggressive cancers, such as glioblastoma and ocular melanoma. The vaccine uses 4 harmless viruses to stimulate the immune response and attack tumors, potentially reducing the need for chemotherapy if approved. Documents for clinical approval have been submitted, and Phase 1 trials are scheduled to begin soon, with up to 48 participants. Russia plans to offer the vaccine free of charge to its citizens if it receives approval. The announcement was made during the 10th Eastern Economic Forum in Vladivostok, following the trial results disclosed at SPIEF 2025 in June. Russia's progress is part of a broader global race, with countries like Britain, Germany, and the US, along with companies such as Moderna and Merck, working on similar vaccines. Cancer remains a major health issue worldwide, with India reporting 874,000 deaths in 2024 and a projected 12% increase in cases over 5 years, driven by lifestyle, diet, stress, and environmental factors. NRG Therapeutics has secured $67 million in Series B funding to advance its neurodegenerative disease therapies. The funding was led by SV Health Investors Dementia Discovery Fund and included participation from international investors and Parkinson's UK. The company is developing NRG 5051, an oral inhibitor targeting the mitochondrial permeability transition pore, or MPTP, which aims to protect neurons by preventing mitochondrial damage caused by toxic proteins. Preclinical studies indicate that NRG's inhibitors can prevent mitochondrial dysfunction linked to toxic proteins like alpha-synuclein and TDP-43, which are associated with Parkinson's and ALS. The new capital will support clinical trials for NRG 5051 in ALS and MND, as well as gather data for Parkinson's disease, with the goal of creating therapies that modify disease progression. Support from the UK's British Business Bank highlights efforts to scale innovative life sciences solutions for neurodegenerative diseases. Emma Johnson from the bank noted this as the 16th direct investment in UK life sciences and mentioned new board members joining to facilitate clinical trial progress. NRG's focus is on developing treatments that address disease progression rather than just symptoms, as neurodegenerative diseases continue to rise in prevalence. Based in Stevenage, UK, NRG has also received backing from foundations dedicated to neurodegenerative research. CEO Neil Miller emphasized plans to expand beyond ALS and MND into Parkinson's and other neurological disorders by targeting biological mechanisms. With recent FDA approvals of genetic-specific drugs for ALS, there remains an urgent need for new therapies. The Series B funding will also support the addition of new board members, including Professor David Dexter of Parkinson's UK, to guide research efforts. Industry experts believe NRG's approach, backed by strong preclinical data, could slow or halt disease progression across multiple disorders, potentially leading to the first disease-modifying therapies for conditions like ALS and dementia. David Baltimore, a Nobel laureate and influential scientist, passed away at age 87 in Woods Hole, Massachusetts. Throughout his career, Baltimore made groundbreaking contributions to virology, including the discovery of reverse transcriptase, which advanced understanding of retroviruses like HIV and impacted gene therapy. He joined MIT in 1968, where his research challenged the central dogma of molecular biology, leading to his Nobel Prize in 1975. Even in retirement, Baltimore remained engaged in scientific research, founding biotech companies and participating in discussions on CRISPR, genome editing, and bioethics. He continued to influence scientific policy, emphasizing the importance of ethical oversight in biotechnology. Baltimore received numerous awards, including the National Medal of Science, and published over 700 scientific articles. He held leadership roles at institutions such as the Whitehead Institute, Rockefeller University, and Caltech, where he raised over $1.1 billion for scientific advancement. As president of Caltech and Rockefeller University, he elevated these institutions' research profiles. Baltimore played a key role in public debates on genetic engineering, bioethics, and biotechnology regulation. He is survived by his wife Alice and daughter TK, and is remembered as a pioneering scientist and leader in biomedical research. Recent research reveals that near-zero index photonics can bridge classical electrodynamics and quantum physics, enabling the transfer of quantum information. This technology supports the development of faster and more secure quantum communication and computing systems. Professor Eric Mazur highlighted that such photonics are crucial for the second quantum revolution, facilitating quantum information transfer. This transition from classical to quantum regimes allows for the creation of more efficient and secure quantum networks. One breakthrough includes the first achievement of long-range entanglement using a compact photonic chip system, which is significant for quantum architecture development. The challenge now is to translate these theoretical models into practical miniaturized systems, potentially as small as consumer electronics. Future efforts focus on experimental validation and the miniaturization of these systems to microscopic scales. Potential applications include advanced lasers, sensitive sensors, and quantum-secure communication systems capable of multi-qubit entanglement. Researchers have also designed a nitrogen vacancy diamond-based photonic chip that can extend quantum entanglement up to 17 times farther than in a vacuum. Additionally, studies on superradiance in near-zero refractive index materials demonstrate the potential for long-range quantum superradiance and scalable quantum technologies. Near-zero refractive index materials enable light to behave uniformly, allowing atoms separated by large distances to interact as if they were close. This facilitates long-range quantum entanglement, a key step toward practical quantum devices. A new cancer drug, STF-1623, has shown promising results in preclinical studies by targeting tumor surfaces to activate the immune system. Developed by Lingyin Li and her team, STF-1623 works by inhibiting the enzyme ENPP1, which normally breaks down CGAMP, a molecule that stimulates immune activation. This inhibition allows CGAMP to accumulate on tumor surfaces and activate the STING pathway in immune cells, prompting a targeted attack on cancer cells. The drug remains bound to ENPP1 for over 24 hours, increasing its effectiveness and reducing side effects, while specifically targeting tumors with high ENPP1 levels. Studies published on September 5, 2025, demonstrated STF-1623's effectiveness in mouse models of breast, pancreatic, colorectal, and glioblastoma cancers, with no adverse effects observed. This development represents the first successful tumor-specific targeting of an innate immune checkpoint, offering a new approach for resistant cold tumors that typically do not respond to existing immunotherapies. Following these promising results, STF-1623 is moving toward Phase 1 clinical trials after receiving FDA approval. The development is supported by organizations including the NIH, ARC Institute, and Angeris Therapeutics, and the drug is designed to work best in combination with other therapies. The research was published in Cell Reports Medicine and highlights an innovative strategy of targeting innate immunity directly at tumor sites. Unlike direct STING agonists, STF-1623 preserves natural CGAMP, potentially leading to more controlled immune responses. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.